Welcome to the Sans Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump into today's deep dive, please take a moment to like, subscribe, and share this with your friends and relatives. It really helps support the Sans Science Experience.
Okay, so today we're plunging into this incredibly intricate world inside your body, the human interactome. Think of it as like the secret language your cells use, this huge network of protein interactions, PPIs.
Yeah,
They basically dictate almost everything.
pretty much every biological function, yeah. How your cells work, how diseases even take hold, it's all down to these interactions. And it's precisely because they're so fundamental that mapping them all out, the human interactome, has been, well, one of biology's grand challenges.
Grand challenges. So every breath, every thought, it relies on these PPIs.
Absolutely. And, scientists figure there are somewhere between, say, 74,000 and maybe 200,000 real human PPIs.
Okay, that's a big range, but still a specific number. It is. But the problem is, for decades, the ways we've tried to find them experimentally, they often give inconsistent results, kind of noisy data.
Noisy.
Yeah, so our databases list over 1 million potential interactions, but honestly, confidence is pretty low for most of them.
Yeah.
It's like trying to listen to a conversation with a ton of static.
Gotcha. So we know this vital network exists, but really seeing it clearly is like finding specific grains of sand on a beach, you said. What makes nailing down the true ones so hard computationally?
Well, just think about the numbers. You've got around 20,000 human proteins, screening every possible pair. That's nearly 200 million combinations.
Whoa, okay.
It's an astronomical task. And another issue, especially for us higher eukaryotes, is we haven't had great data for coevolutionary analysis.
Coevolutionary analysis, like seeing how proteins evolve together.
Exactly. You try to spot partners based on subtle evolutionary changes, but if you only have a few snapshots in time, it's incredibly difficult to see those linked changes.
Right. So facing this huge challenge, these researchers didn't just chip away at it. They came up with something new. Two big innovations, I read.
They did. They sort of reimagined the whole approach, cracking open two major bottlenecks. The first breakthrough was about boosting those coevolutionary signals.
Boosting them. How?
Instead of just using the existing protein databases, which can be incomplete, they did something, well, frankly, audacious. They mined 30 petabytes of raw, unassembled genomic data.
30 petabytes. I can't even picture that much data.
It's enormous. And it was from over 21,000 different eukaryotic species, focusing especially on chordates, and arthropods are evolutionary cousins, relatively speaking.
Wow. Okay, so from this ocean of data, what did they do?
They built a really smart pipeline to directly assemble the protein coding sequences from that raw data. This created what they call OmicMSAs, multiple sequence alignments, derived directly from omics data.
OmicMSAs. And these were better than the old way.
Much better. Think of them as super detailed genetic blueprints, way richer in evolutionary history. About 7 times deeper, they calculated.
7 times.
Yeah. It's like turning up the volume on really faint signals. Suddenly, those subtle coevolutionary clues that hint at an interaction became much easier to spot.
Okay, so that's the data foundation. Incredible data. But you still need to read it, right, to actually predict the interactions.
Precisely. And that leads us to the second big innovation, a new AI model.
Ah, the AI component. Makes sense.
They developed the new deep learning network. It's called RF2 TPI, and it's built on the RoseFold architecture.
Rosettefold. I've heard of that. It's known for predicting protein shapes, right?
That's the one.
Yeah.
Very powerful. But to train this version, RF2 PPI, effectively for interactions, they needed more than just the known PPIs, which is a pretty small data set, actually.
So how did they bulk up the training data?
They created this massive distillation set. Basically, they taught the AI to recognize interaction patterns by showing it a huge library of how protein bits or domains partner up.
A distillation set. What was in it?
Get this. 1 million domain interactions, DDIs. They pulled these from 200 million predicted protein structures in the AlphaFold database.
Wow. Using predictions to train predictions. That's clever.
It is. It's like teaching the AI to see how millions of protein puzzle pieces fit together in 3D, based on AlphaFold's best guesses.
So you combine this supercharged genomic data, the OmicMSAs, with this cleverly trained AI, RF2-TPI. What happens when you point it at the human interactome?
The results are pretty remarkable. They systematically screened 191 million human protein pairs. And from that, they predicted 18,316 PPIs with an expected precision of 90%.
90%. That's high confidence.
Very high. And here's the really exciting part. 5,578 of these predicted interactions are completely new.
Over 5,000 new connections.
Yep. And many of these novel ones involve transmembrane proteins, TMPs. These are proteins stuck in cell membranes, notoriously hard to study with wet lab experiments.
Ah, because they're difficult to isolate and work with.
Exactly. So this computational approach really shines there. And these predictions, they come with 3D models too. It nearly triples the number of human PPIs, where we have good structural information.
That's huge, tripling the structural data. So, okay, this is clearly a big deal for researchers, but
Yeah.
bringing it back to our listeners, why does mapping this interactome matter to you? What's the real-world impact?
Well, this detailed map gives us critical insights into, not just how proteins normally work, but how diseases mess things up. It gets at the mechanisms.
Mechanisms, like how a genetic mutation causes a problem.
Precisely. For example, among the new predictions, they found interactions involving GPCR's G-protein-coupled receptors. These are huge targets for drug development already.
Okay.
So now researchers can investigate newly predicted interactions, like one between GPR143 and HPS1. Both are linked to albinism, found in melanosomes. Seeing how they might interact could open up totally new ideas for therapies.
Wow, so directly pointing towards potential new drug targets or strategies.
Potentially, yes. Or think about immunity. They predicted an interaction between IRTM1, which helps fight viruses, and VMP8, involved in basically cellular waste disposal.
Okay.
This suggests a completely new way IITM1 might be blocking viruses from getting into cells. It's opening doors to understanding fundamental processes.
It really sounds like this work isn't just adding entries to a database, it's providing a much, much clearer blueprint for how life actually operates at that molecular level.
That's a great way to put it, a higher resolution blueprint.
And thinking ahead, could this knowledge let us, I don't know, eventually engineer these networks? Not just treat diseases, but maybe prevent them by tweaking these interactions.
That's the long-term dream, isn't it? Precision medicine taken to the network level. They're not there yet, obviously, but this kind of comprehensive map is a massive step in that direction.
An incredible leap, truly. It really shows the power of combining these huge data sets with sophisticated AI to unravel biological mysteries. Well, that's all the time we have for this deep dive. Thank you for joining us. Please don't forget to like this video and subscribe to the Sans Science Experience for more exciting science explorations.